Hi everyone, welcome to my video presentation. My name is Momcilo Prodanovic and I'm a PhD student at University of Kragujevac in Serbia and Research Associate at BioIRC Research Center. I will talk in my presentation about the work that we've been conducting on our SilicoFCM Horizon 2020 project, which is analyzing heart muscle twitch experiments in mice, rats, and humans and simulating the intact cardiac muscle responses using our in-house built platform Musico. The main motivation for this work emerged from the challenges researchers encounter while trying to understand molecular origins of human cardiomyopathies. Classical experimental approaches were limited due to lack of available human cardiac tissue, both healthy and diseased. Even though there is abundance of data available in more convenient mice and rat studies, it proved hard to directly translate the findings to humans because of the differences in sarcomeric protein characteristics and contents. Moreover, scientists and researchers found it's an enormous challenge to integrate observations from multiple experiments under different experimental conditions, setups, and scales. For example, whether if it's a protein-to-protein interaction in assays, single myofibrils, muscle fibers, or whole organ experiments. One solution for translating data between species and better understanding human cardiomyopathies is the use of detailed computational models of sarcomeric functions. Before we get into computational modeling, I'd like to show you one typical muscle experimental setup for measuring forces, changes in sarcomer length, and calcium transient during twitch contractions, which is in the left of this slide. The middle image shows how the muscle fiber looks like under the light microscope, where the darker lines represent M lines in 3D sarcomer lattice, with mostly thick filaments or myosins, while lighter grays are Z-disc regions with the thin filaments or actins. There are also many other muscle proteins involved in 3D sarcomer lattice, like troponin, tropomyosin, myosin-binding protein C, nebulin in skeletal muscle, and others which are not shown in this slide. Now, how can we model muscle behavior? For that, we developed a computational tool called Musico, which stands for Muscle Simulation Code. Musico is a modular computational platform for simulating a wide variety of experimental protocols that are often performed on muscle fibers or cells. It contains various thin filament regulations, actomyosin cycles, explicit 3D geometry of sarcomer, and incorporates spatially randomly distributed mixture of myosin isoforms and structural features specific for each of the species. This all makes Musico platform well suited to be used as a tool to translate data between mice and humans. On the next slides, I will explain in detail each segment of Musico platform used in our simulations. As you probably know, muscle contraction and relaxation are regulated by the calcium-dependent azimuthal movements of tropomyosin troponin complexes over the surface of actin filament. Structurally, tropomyosin covers 7 monomers on one strand of the actin double helix, which is shown in the bottom left panel and is associated with one troponin molecular complex consisting of troponin T, troponin C, and troponin I. Calcium regulation is defined by calcium binding or dissociation from cardiac troponin C, which changes the affinity of T and I to actin. This is shown in 4-state cycle with closed and open allosteric states. Closed states are painted in red and open in blue. The equilibrium rate constant of calcium binding to T and C closed states is Kca, which is defined with the forward constant rate that linearly depends on calcium concentration and calcium-independent dissociation constant. The dissociation constant of T and I from closed to open state is defined by a first-order affinity lambda, which is attenuated by epsilon 0 in top pathway without bound calcium. Actomyosin cross-bridge cycle used in Musico simulation is updated minimal 5-state cycle shown in red square with 3 attached and 2 detached states, where the attachment rate of myosin heads to actin sites is regulated with calcium as shown with arrows. In addition, we included a 6th state called park state, in which the two myosin heads self-associate and pack down onto the thick filament. Park state is regulated by calcium, and it is necessary in order to keep the binding flux of myosin heads low when calcium concentration is low during the resting phase of muscle contraction, while allowing more myosin heads to be available for binding once the calcium concentration is high. Park state is defined with backward rate K minus PS, which is calcium independent, and forward transition rate K plus PS that depends on calcium concentration via sigmoidal equation, where KPS0 is baseline rate, KPS max is the amplitude, B is the Hill coefficient, and Ca50 is calcium concentration at 50% of KPS max. Musical simulations contain complete lattice with myosin, actin, and titin, shown in panel A. 3D lattice in axial direction is typically used in all available models, while in Musico we account for myosin interaction with actin in 3 dimensions, shown in panels C and D. This is because myosin crowns are shifted for 40 degrees, so some are aligned and some are not with actin filaments. We also included variable length of thin filaments in our simulations. The filament lengths in each subsystem are randomly determined by Monte Carlo simulations using probability density function from experimental measurements of thin filament lengths in rat atrial trabecula, presented as gray bars in panel A. Best fit of thin filament length distribution, which is green line, and cumulative probability function, which is the red line in panel B. The nonlinear elasticity of titan molecules is calculated in Musico using the experimental curve of Helms et al. Observed in cardiac muscle, which is shown as black symbols with bars in panel C. In the same panel, the red triangles are estimations for wild type from Cazorla et al. At 2 sarcomer lengths. The effect of different sarcomer and thin filament lengths simulated with Musico in isometric conditions shows that along with changes in lattice spacing, the average overlap between thick and thin filaments is changing, which affects the effective binding rate as well as active and passive tension. In order to simulate twitch responses in trabecula with Musico, we included serial elastic element at the end of the contractile element consisting of multi-sarcomer muscle fibers. The origin of trabecula series elasticity is quite complex and might include muscle fiber alignment and connective tissue serial elasticity as shown in a rat ventricular trabecula in left panel. The highest magnification shows stratifications along muscle fibers as well as imperfect alignments and gaps between and along muscle fibers. Karemani et al. Measured tension sarcomer displacement relation during isometric twitch contraction of rat trabecula, which helped us estimate stiffness of trabecula serial elasticity. The orange circles in the right panel represent the tension displacement loop, which shows higher forces during sarcomer shortening and lower forces during sarcomer lengthening. The arrows signify direction of these changes. For simplicity, we derived a nonlinear serial spring-like elasticity from average force of force displacement loop represented as gray line, which is used in all species in our Musico simulations. Lastly, it is important to mention that we can distinguish between fast alpha and slow beta twitch muscles related to fast and slow interactions of myosin with actin. In different types of muscles and species, the proportion of these myosin isoforms vary. Thus, we included this ability in Musico platform. The fast and slow twitch data from mouse skeletal muscles reported by Nurhusen are fitted with Musico simulations in top panels. Calcium transient was the same and had a peak of 1.6 micromolars at 6 milliseconds and decayed to a low calcium concentrations at about 35 milliseconds. In the bottom panel, we were changing the percentage of fast and slow myosin isoforms in sarcomers to see its effect on twitches, which shows slower rise and relaxation times as well as lower generated force as the percentage of slow myosins increases. With the same fraction of myosin isoforms, Musico predicted 4 PCA relations. In panel A, the magnitude of force illustrates coupling between number of bound cross-bridges reflected through the force and cooperativity between bound myosin and regulatory units. Normalized force PCA graphs are shown in panel B, which better illustrate the increase of calcium sensitivity and Hill coefficients as the percentage of slow myosin isoforms rises. Finally, using Musical platform, we simulated twitch transients in intact trabecula from mice, red lines, rats, green lines, and humans, blue lines. Experimental twitch traces, presented as dashed lines in top left panel, were taken from Chang et al. For all three species, while Musical simulations are displayed as full lines. Changes in sarcomere length during twitch contractions, presented as displacements in our graph, were not recorded during the experiments, thus we only showed here Musical predictions. Simulations were performed at 25 degrees Celsius and sarcomere length at 2.2 micrometers, as it was stated in Chang et al. We changed the percentage of myosin isoforms in species, approximating the experimental findings, thus having 100% alpha in mice, 100% beta in humans, and combination of 75% alpha and 25% beta isoforms in rat simulations. The top right panel shows that along with different myosin fractions, these species have different calcium transients, which were estimated from various experimental sources. Interestingly, the top left panel shows a good reflection on speed of the heartbeat between species, where mouse heart is about 5 times faster than humans. This table shows musical parameters that were different between species. The excellent fits of trabecula twitches were achieved with changes only in crossbridge cycle parameters. In case of ADP release rate and hydrolysis forward and backward rates, values were assessed from the kinetic analysis of proteins in solutions, while myosin actin binding rates were estimated by fitting simulations to the experimental tension transients and reflect the slowing from alpha to beta myosin isoforms. In conclusion, using musical simulations, we demonstrated that differences in myosin isoforms can lead to the observed behavior in intact cardiac muscles during twitch contractions in mice, rats, and humans. We also showed that the twitch kinetics are tightly coupled to the calcium transient specific for the species. As I already said, the experiment fits of trabecula twitches were achieved with changes only in crossbridge cycle parameters, which some were assessed from the experiments in solution. However, threefold slower rate of myosin binding to actin for beta isoform in humans than in rats could be in part exaggerated because the transition from park state was kept constant in all simulations. We are currently working with our partners on obtaining one complete dataset for all three species under the same experimental conditions, including intracellular calcium and change of sarcomere length in order to validate these presented findings. Finally, I would like to thank everyone in my team for helping me in this project, and of course, European Union for funding it.